Good morning. Thank you very much for coming out this morning for the last in our symposium series on World War I. We'll start this morning with Dr. Pearcy's lecture on the influenza pandemic of 1918, and immediately following, Sal Buffo will speak on post-traumatic stress disorder and trench warfare. We will take a brief intermission, and then our master chorale will be performing at 1 o'clock. So thank you very much. We have a full morning. Before I leave the podium, I'd just like to remind everyone that Yavapai College does hold the national roll call for Veterans Day. We will be here from 7:30 until all names of our fallen veterans have been read. So from 7:30, wait, I have it right in here, 7:30 in the morning until 5 o'clock, we will be in this room reading off the National Roll Call Monday. So if you're interested in joining us for that, we'd appreciate it. So, yes. Yes, ma'am. Yes, ma'am. Thank you for asking. And so, thank you very much for coming, and please welcome Dr. Matthew Pearcy.
So is this thing working? Okay. So thank you very much for being here. You guys are choosing to come in on a beautiful Saturday morning and hear all about infectious disease and death. I'm not sure what that says, but thank you very much for being here. So what I'm going to talk about today is influenza. So my background is basically infectious disease. So this is something that I find extremely fascinating. And one of the things that's fascinating about it is how it kind of affects the history of things as they happen throughout American history and history in general. So what I'm going to do today is mostly focus on influenza virus. The story kind of starts in World War I as far as Americans are concerned, but the story of that virus pretty much continues to present day. So we're going to spend a little bit of time in World War I, and then we're going to kind of talk about what's happened since then and what we've come to learn about that virus just in the last few years. And also, there's a lot of science packed into this talk. And so what I want to do is have it be very informal. So if something doesn't make sense, someone please pop their hand up, and we'll clear anything up. I have one student in here, and that's Angel. So Angel, if something doesn't make sense, you let me know. Okay. So to start this off, talking about influenza, I want to talk in very, very broad, basic terms, because I think in society in general, there's kind of a confusion about the different microbes that can infect us. And depending on what type of microbe you're infected with, it has huge implications on what's going to happen to you and also how you're going to be treated. So to start this off, we're going to talk first just very briefly about the two major families of microbes that make us sick. Now, there's more than just these two we're going to talk about. You can only see one, but there's going to be two. Different fungals or different fungi can infect us, so we can get fungal diseases like valley fever. Some people have probably heard of that. It's endemic in this area, especially in Phoenix. The second major family of microbes that can infect us are the bacteria, right? So here I have a picture of just two different bacteria. On top is E. coli. Pretty much everyone's heard of E. coli. Right now, every person sitting in this room, there's E. coli in your large intestine making vitamin K for you. If they weren't doing that, your blood wouldn't clot, right? So some E. coli are very good. But everyone's also heard about some of the E. coli outbreaks that happen because different kinds of bacteria can get in our food, different kinds of E. coli can, and they can make us sick. So there's some good E. coli, there's some bad E. coli. This picture down here is a picture of Staphylococcus aureus. So people hear in the common news about staph infections, about MRSA. That's a specific type of Staphylococcus aureus. There's many different bacteria that cause infections. Most of our chicken swimming in Campylobacter jejuni, which is a bacteria that can give you food poisoning. That's why you have to cook chicken so well. Ulcers can be caused by a bacteria called Helicobacter pylori. So there's many, many different kinds of bacteria. The good thing is we can treat them with antibiotics. Antibiotics, that word means anti-life. And so when we take antibiotics, they kill the bacteria, and that helps us get better. So antibiotics kill bacteria. We can contrast that with the viruses, which is what we're going to focus on today. Antibiotics are 100% useless against viruses. You can take as many antibiotics as you want, as many different types as you want. And if you're infected with a virus, they are going to do nothing. Okay? There's some antiviral drugs out there. There's drugs to help treat HIV, which is a viral infection. There's a couple medicines out there to help treat the flu. The general consensus is they're not really very effective. And they're not antibiotics. They don't actually kill the virus. They just help you get through the infection. It wouldn't be inaccurate to say that every time one of our cells gets infected with a virus, that cell is dead. So antibiotics do not work when it comes to virus infections. So if we look here, this is a picture of HIV. That's a virus. This up here is a picture of flu. That's what we're going to talk about today. There's many other examples. The common cold is caused by a rhinovirus, right? Rabies is caused by a virus. Polio is caused by a virus. So when you hear about these different illnesses that are caused by these different organisms, it's really important to know what they are. Are they a bacteria or are they a virus? Because that has huge implications on how you're going to get sick and if you're going to be able to be treated. So we're going to leave that behind and move on to the viruses. Okay? The other thing about viruses that's really interesting to me, and it also differentiates them from bacteria, is that viruses are not alive. They can kill us, but they're not alive. So if we look at bacteria, everything we associate with being alive, taking in nutrients, right? Making energy, making new proteins, moving around, bacteria can do that all on their own. If you had a little jar full of bacteria, they could swim around just fine on their own, reproduce just fine, because they're alive. Viruses are not alive. They don't make their own energy. They don't reproduce on their own. They don't move around the environment. They're basically just a little tiny organism that's dead. The only way that they can actually reproduce is if they get inside of us. So that's a huge difference between the viruses and the bacteria as well. Okay? The last thing that makes them so different is size. When you start talking about nanometers, like billionths of a meter, it's really hard to envision that kind of time scale. So for me, I like to think about it like this. We're human beings, right? And so all of us are different sizes, so we're going to have different amounts of cells that make us up. But in general, each one of us is made of trillions of cells. So if one of our cells was as big as a professional baseball team's playing field, like Bank One Ballpark, the entire ballpark, if one of our cells was that big, then a virus would be about as big as the entire pitcher's mound. Or I'm sorry, a bacteria would be about as big as the pitcher's mound. But these little non-living viruses would be as big as a baseball. So you compare the size of a baseball to an entire ball field, and you get the sense of just how tiny these guys are compared to us. They're extremely simple, but they're extremely deadly. So that's kind of the intro on viruses. Flu's a virus, so all those things I just talked about that differentiate them from bacteria is going to hold true for the rest of this presentation. So to talk about this, the influenza virus, I want to put this in the context of just how much effect it had on World War I. And I think the best way to do that is with some numbers. So there's a lot of data on this slide, but what I really want to draw your attention to is what's specific to us overall. So if you look at America's involvement in World War I, we had a little over 4 million people that were mobilized to fight in the war. Of those, 116 died. This slide here is a little misleading. It says killed. It should say died. So we'll talk about what that means in a second. So about 116,000 people, American soldiers, died in World War I. And if we go down here to the very bottom, the other number we care about is that if we look at all the soldiers that died, all sides, everyone combined, it was a little over 8 million killed. Okay, so if we want to put World War I in the context of other American wars, then what we're looking at is right here. So of those little more than 4 million soldiers that were mobilized, we know over here that about 116,000 died. But if we look at how they died, 53,000 were killed in the actual battle by shrapnel, gas, all that kind of thing, bullets, whereas 63,000 died from accidents and disease. And the vast majority of those were disease. So more soldiers died in World War I from influenza than ever died in combat. And that's true in World War I, and if you look backward in time, that's true in most wars. If we look in the Civil War, 140,000 killed, but 250,000 or actually died of diseases and accidents, and most of those are disease. So if you look at some of the wars we've had over time, it's common for more people to die from disease than ever die in combat. So in some ways, the story of wars is actually the story of infectious disease. Granted, I'm biased when it comes to that kind of thing. So if we want to compare those numbers to what happened with influenza during basically 4 months in 1918, we can compare those numbers to these. So in America alone, there were almost 700,000 people that died of influenza in basically almost 4 months. Okay? If we want to look worldwide, the estimates really vary. People used to say around 50 million people died of influenza. Now, depending on who you ask, that number is being revised upward to maybe 100 million. So these are incredibly large numbers of people who were all killed by a virus. So there was this catastrophic war that killed a lot of people. But during that war was this sickness that killed many more people than ever died in actual battles. So when we look at this, the soldiers were just decimated by the influenza virus. And so where I want to go next is to talk a little bit about World War I, basically how influenza spreads and why the soldiers were so susceptible to it. Okay? So the first thing I want to talk about, it wouldn't be a proper infectious disease talk without some disgusting things being talked about. So when people sneeze, right, a good, healthy, average sneeze has about 40,000 droplets, okay? And an average sneeze can travel at least 50 feet. So if I sneeze during this talk, everybody, right? So in one of those 40,000 droplets that comes out in a sneeze, there can be easily 10,000 flu viruses in one droplet. And you're spewing out 40,000 of them. So to continue the math a little bit, influenza or flu, I'm just going to call it flu for the rest of the talk probably. Flu is one of the most easily spread viruses that there is. If you're going to get infected with something, if you get infected with one flu virus, one Ebola virus, you're probably not going to get sick. You need a certain amount of organisms to get in your body. You need what's called an infectious dose. And so to get the flu, you need anywhere from about 100 to 1,000 viruses to enter into your body. Okay? That's enough to make a healthy person sick. Okay? So 40,000 droplets, 10,000 in each droplet, and you only need 100 to 1,000 to get infected. And a sneeze can go 50 feet. So you put all that together, and you can see why influenza is so infectious. And when people sneeze, you know, there's like, we're going to continue the disgustingness of the morning. There's large droplets, right? And there's little tiny ones. They're all sizes. And so if I sneeze in this room, then a lot of those large droplets hit the floor immediately, but some still kind of float around. So you come in here maybe 20 minutes later, it's entirely possible you breathe one of those in and you're sick, right? And when I sneeze out all these viruses, they land on surfaces. And depending on what kind of surface it lands on, you can really have a large difference in how long the virus lives outside the body. If it lands on paper, it's going to live for like 15 or 20 minutes, right? So you go to the store, right? You're in line to check out, and someone sneezes, hands the person some money, and then you're behind them in line, and you get that money as change. That's a really easy way to get infected with flu. If you have a hard surface, like that's non-porous, like metal or plastic, especially if it's cooler in temperature, it can live for hours, maybe even days. So if you were, you know, someone did something to get flu here, and then I touch this, and then I touch my mouth, now I'm infected, okay? So flu is extremely contagious. It's very, very easy to spread from one person to the other. Respiratory droplets, surfaces of things, if you touch those things and touch your mouth or touch your eyes, then you're infected too, and you pass it along. Another thing that's kind of it makes it hard to keep people from getting infected with flu is that flu is actually a disease that we spread before we're sick. Ebola's not that way, thank God, right? You listen to the news nowadays, if people show symptoms, then they can spread the disease. If they don't show symptoms, then they can't spread it. Flu's not like that. About 1 day before you actually start showing signs, you're contagious. You're shedding virus into the surroundings. So you can get people sick and not even know it. Okay? And some people get flu, don't get sick at all, but are spreading it in the environment, right? So this virus is extremely contagious. It's extremely easy to get. And look what it looks like when you got to send 400 or 4 million people across the ocean to war, right? People are on a cruise ship in their own cabin, right? These people are crowded on these decks, sleeping one on top of the other, and that's a perfect recipe for the virus to infect one person after another. Because if this person feels great, he could already have the flu. So by the time he gets sick and you quarantine him in a sick bay, he's already spread it to these guys. And so that just spreads and spreads. If we look at what people tried to do back then, this is where we're going to talk about what we know now compared to what we know then. Because it's always interesting to me to put infectious disease in the context of their time. Okay? So if you look at 1918, we knew there were bacteria. Okay? We could culture them. We could see them. We didn't have antibiotics yet, so we couldn't do much about them. Right? But people knew by the onset of World War I that bacteria were present. They could cause sickness. We didn't have antibiotics, but we knew to keep things clean. So unlike the Civil War, surgeons would wash their hands before they actually performed surgery on someone. When they were setting up a military camp, they wouldn't have the latrines upstream from where you were going to drink the water. So a lot of stuff about being aseptic and preventing the spread of bacteria that way, people were aware of those things. So even though there weren't antibiotics yet, those didn't come till World War II, we still knew how to do things to prevent the spread of bacteria. But I told you viruses are tiny compared to bacteria, right? So they also knew in 1918 that sometimes things made people sick and we can't find them. They would try to grow them up in something like they were used to growing bacteria and nothing would grow. And then someone discovered how to make a filter that had extremely tiny pores. Those are just holes in the filter, like a screen, right? And so when you would pour a solution from someone who was sick through this filter, the holes are so small that it would trap the bacteria, right? But viruses are so tiny, they could make it through the pores. And then so scientists would take this fluid that they knew didn't have bacteria in it, and they knew there was something in it that could make people sick, but they didn't know what it was. Okay? So they knew there was something besides bacteria that was making people sick, but there was no idea what it was. It wasn't until the late 1930s when they actually discovered right in front of their own eyes using electron microscopy the very first virus, tobacco mosaic virus. It infects plants. So it wasn't until the late 30s that we could actually see these things. So everything we knew about viruses was very, very little. We knew they were there, something was there, but that was it. So people tried to protect themselves as best they could. This is a boxing match going on a ship that's going to head off to war to keep people entertained. And so if you look here, people are wearing masks. See all these masks right here? Except for this guy. He's pretty confident that things are okay. So the one thing they could do to try to prevent the spread was wear these masks that had pores that were much bigger than the viruses. So they weren't effective really at all. They could maybe help a little bit. If someone sneezed right in your face, it would keep some of the large droplets off of you. But given how much the flu spreads and how long it hangs out, this kind of mask to prevent the flu would be pretty much completely ineffective. Okay? If you look here, this is soldiers sleeping here. And there's actually a picture I found that was before this, and all the beds were there, but none of the screens were there. And so the way they had to deal with it back then to try to help prevent the spread in sleeping quarters was put these screens around, which is the best they could do, but pretty much completely ineffective. The flu is so contagious that everything they could do to prevent its spread wasn't effective because they didn't know what to do because they didn't know what they were facing. Okay, and so once people got to the front and started to actually fight, some of the conditions in war made it so they were even more susceptible. Did someone just cough and sneeze? Out. I'm just kidding. So, I'm just kidding. Someone's got flu or Ebola. I don't know what we're going to do. So, okay. Okay, so if you look at these soldiers here, it's obvious they're stressed out, right? So I can't even imagine what it's like to be in a war. I mean, Amy gave a presentation about the causes and the consequences, and Gino gave one on gas warfare and all that. But these guys basically entered a war where the first time they were the widespread use of machine guns, right? And they started off on horses, at least initially. So they go piling over the trench and getting mowed down. They knew that was going to happen. They're living in like just complete squalor and under tremendous stress. And so bringing that back to the science part of it, stress is a really good thing, right? Like if you need to fight or you need to run or you need to take care of something stressful immediately, your body floods with hormones that brings more oxygen to your brain and your muscles, and you shut off blood supply to your kidneys and your reproductive system because you don't need those to fight off a lion, right? So stress is good unless stress lasts a long time. If stress lasts a long time, regardless of the reason, that also starts to play with your hormones. And it does a bunch of different things, but one thing it does is it raises the level of a hormone called cortisol. And if you have high levels of cortisol in your body for a long time, like for weeks or months, if you're in a trench fighting someone, what the cortisol does is basically turn down or turn off your immune system. So long-term stress, regardless of the reason, psychological, physical, or whatever, it's going to shut off your immune system. So these guys are fighting a war in the midst of this virus that's incredibly contagious, probably more contagious than any other pathogen you could think of, or at least as contagious, and their immune systems aren't functioning. So the soldiers really don't have a chance, right? If the war doesn't get them, in battle, then there's a good chance they're going to fall sick from this flu that's going to most likely kill them as well. So that's what's going on in World War I. So what I want to talk about now is the virus itself. So I want to talk about some virology. So I told you how tiny viruses are, right? This is a picture, this is an electron micrograph of influenza viruses. So whenever you look at something under the microscope, like they should always tell you how much it's been magnified so you have a sense of how small it is. This picture is magnified about 100,000 times. So regular microscopes, like the kind that we use like in the teaching lab here, they'll magnify things about 1,000 times. That's as much as they can get. This is 100,000 times. I would love to be able to teach with one of these, and as soon as I can convince the administration to spend 50 grand on an electron microscope, we'll hook you up. I wouldn't hold your breath. So this is a picture of the virus, right? And so during World War I, people didn't have a picture of this virus. They had no idea what it was. And it's actually an interesting story about how things move forward, because when the war was over, the flu disappeared and people didn't want to talk about it. If you search through the history, like the autobiographies, these people who were involved in the response to the flu and the war and all that, they rarely mention the flu in more than 1 or 2 paragraphs. Like even though this was a huge thing that killed 50 million people at least, people back then didn't want to talk about it. And the theory is it was so horrific that people just wanted to put that sickness and put that war behind them and just move on. And if you look at the literature, it's really interesting how little there is from people who were involved at the time. So the story of how we actually found the 1918 flu in modern days is a really interesting story. This isn't a picture of the 1918 flu, but we actually have the 1918 flu now. In a freezer somewhere. What happened was, in the early 50s, this guy named Johann Holten, I think I'm pronouncing his last name correctly, he came up with the idea that if he went way up north to North Alaska where the permafrost is, and permafrost is basically ground that never thaws, right? So his idea was, I'm going to go up north to where the permafrost is, and I'm going to dig up people who died of the flu, and I'm going to get the virus. And it's a crazy story. He submitted a grant to the government to get some money to do that, right? And they didn't call him back for months. And then he actually had a friend who was in Congress, and he's like, what the heck is going on? And so the congressman went back to the people who gave out the money and was like, why aren't you funding this guy? And it turns out that the government had taken his idea, and they were planning their own trip. So the US government went there to try to find bodies in the permafrost with the flu, and then this guy did it all on his own, and in 1951, no one had any success. They couldn't find the flu. And then you flash forward to the mid-90s, there was a different guy named Jeffrey Taubenberger. He was also a researcher. He wanted to find the flu too. And what he did was look in archives for medical specimens. So during World War I, if someone died of something funky, what they would do is they'd take a chunk of tissue, put in parafilm, throw it in a box. There's like literally millions of samples like that back east in different places. And he found, actually, in some of those parafilm blocks, some tissue from people who died of the flu. And it took him years, a couple years, but he actually found fragments of the virus. And so he started to reconstruct it from that, got it partially reconstructed, and then ran out of material. And when he ran out of material, he published what he did have. And then the guy from 1951 was still alive. Johann was still alive. So Johann got in contact with Jeffrey and said, hey, I know where you can probably find more 1918 flu. And so they went together to the permafrost, way northern Alaska. And they ended up finding a woman who had died of the flu, and the flu virus was still partially intact in her. And it was intact enough that they managed to figure out the entire structure of the flu. So now you can go online and you can look at the entire genetic structure of the 1918 flu. So we know now exactly how it was made up. But it took until late 1990s, early 2000s to get all that figured out. But people from the 50s helped. So it was kind of a crazy story. So to talk about how the flu spreads, we got to talk about the virus structure, the important parts of it. So that's this picture here. So this is just a very stylistic rendition of influenza virus. Okay? So if we look here, here's what's important. This is a very tiny structure, right? On the surface of it are all these proteins sticking out. So here's one here. It's called hemagglutinin. This is the purple guy. And there's also one that's this greenish color. This is neuraminidase. Okay? These are just proteins on the surface of influenza virus. So you read in the popular news, you hear about H1N1, H5N1. Does that ring a bell? If we look at all the different flus, there's a bunch of different kinds. We know of at least 16 H's, or 16 hemagglutinins, and we know of at least 9 different neuraminidases. So H1N1 just means that virus has the hemagglutinin we name number 1, and the neuraminidase we name number 1. It's just a classification scheme to keep track of which viruses are which. Here's what's most important about that, though. Hemagglutinin is how the virus gets into our cells. It's a key, right? A virus just can't land on any old cell and get in. It has to have a receptor, which we can think of as being analogous to a key. It has to be able to come up to one of our cells, stick its key in a receptor protein on the surface of our cells, and that's what gets it in. So if the hemagglutinin can't bind to our cells, it can't get in. Okay? So to get into our cells, hemagglutinin has to be able to bind to our cells. When the virus gets into our cells, that's the only time it can ever reproduce. We'll talk about that in just a second, but viruses are dead, right? So the only way they can make more copies of themselves is if they get into our cells. So once one influenza virus gets into our cells, it forces our cell to make maybe 1,000 viruses. Then they have to get out so they can go spread to other cells. And what gets the virus out is neuraminidase. Okay? So those are the two important kind of proteins we're going to talk about. The other important part is this right here. This is the genome. So the genome of flu virus consists of 8 different segments that provide the entire genetic instructions to make all of this. Here's the analogy I like to use. So if you think of this whole structure as being a house, the genome is the blueprints on how you make that house. Okay? If you look at us, like we have cells, right? People have probably heard of chromosomes. And each one of our cells is chromosomes. All they are is blueprints on how to build us. So inside of flu, you have basically 8 pages of blueprints. That's how I kind of want you to think of them. The virus is going to use those as a set of instructions on how to build itself. Okay? The important part of this is if changes happen in these blueprints, the blueprints are being used to build the virus, right? So if you change the blueprints, you change the whole structure of the virus, right? If you make a little tiny change to one little part of a blueprint to build a house, maybe you move the window like 3 or 4 feet to the left. Little minor changes, right? But what if you tear out an entire page and replace it with something entirely differently? That's going to have huge changes in your house. And genetically, that's what happens here. If we have little tiny changes in one blueprint, the virus is going to be different, but just a little tiny bit, right? If we change an entire blueprint page here, that's going to have huge consequences on what the virus is going to look like. So that's going to be really important. Okay. Angels, this makes sense? Okay. This is like key to everything. Does this make sense? I'm sorry, the only person I really know their name, so I got to call on you. Okay. Okay, so this is our flu virus. The H is going to get it in to our cells. The N is going to get it out. Any changes to these blueprints are going to result in changes to the overall virus. Okay, so we're going to build from there. So this whole structure is way convoluted. We don't care about the details. What we want to talk about is big picture. So if you look at this entire structure here, this is one of our cells, right? This is the nucleus of it where our DNA is. So we have a trillion cells, trillions of cells, all doing things for us, making things we need like insulin, just going about their lives. Nerve cells are sending information from one part of the body or the other. Muscle cells are contracting. We have tons of different kinds of cells. The important part of this is what happens when flu comes along. So this is our flu virus, right? Here's the H and N proteins. Here's its 8 little pages of blueprints or its genome. It binds to the outside of our cells, gets in, and the take-home message from this thing is once the flu virus is inside of our cells, it hijacks our cell. Our cell stops doing what it's supposed to be doing and starts spending all its time and resources making new viruses. Okay? And it's fast. Flu is so fast. With flu, 6 hours later, your cells are spewing out influenza viruses because they've taken over the machinery of your cells. That's what all viruses do because they're not alive, right? So how could they ever reproduce? They reproduce by getting inside of our cells, hijacking our cells' machinery, and our cells start taking all their time and resources, not doing what they're supposed to be. They start spending all their time and resources making new viruses. So maybe one virus comes in, 1,000 viruses leave, cells dead. Those 10,000 viruses or those 1,000 viruses infect 1,000 more cells, get inside, make 1,000 each, and you see how that would spread, just horrifically quickly. So when this virus gets inside of our cells and takes over the machinery, starts making new viruses, we feel that, okay? And so what we have are some of the symptoms of the flu. Right? And these are all things people are aware of. Some people get a headache, some don't. Some people get fevers, sometimes really high fever, sometimes people don't. It just depends. You get muscular tiredness, coughing, runny nose, vomiting, aches and pains. These are all indicative of the flu. They're also indicative of a lot of other sicknesses, right? The flu kind of has symptoms like that. Ebola has some symptoms that are somewhat similar to that. The interesting thing about these symptoms is a lot of them are caused by our own body. So when you get infected by a virus, your body starts making these little compounds called cytokines, or maybe some people have heard of interferon. These are all compounds that help us kill invading viruses. They also make us feel sick. So what makes you feel sick when you get the flu is not the virus, it's your own immune system, okay? Which is an interesting thing about the 1918 flu. Flu does this crazy thing where sometimes it kills certain age groups more than others. What usually happens when a population gets infected by a microbe that can kill people is it kills the very young and it kills the very old, right? And if you're like 15 to 35 or 15 to 40, you usually have the best chance of being okay because you're young and your immune system is at its peak, so on and so forth. The 1918 flu was completely different. It preferentially killed young people. So people who were like 15 years old to 35 years old, the mortality rates were the highest in those people. And it almost would leave the really young people and the really old people who are usually the ones who die the most, they were the ones most likely to be okay. And the reason for that is thought to be the person's own immune system. So I told you these compounds that we make actually make us feel sick, like interferon, other cytokines, things like that. But in the 1918 flu, young people who had the strongest immune systems, they would release so many of those compounds, they would create what's called a cytokine storm, or basically their own immune system made so many compounds to fight the flu that their own immune system killed them. So that's really unusual in a disease. And flu does different things like that as well. So in 2009, there was an outbreak, a worldwide pandemic. Huge amounts of people got infected by flu in 2009, but not that many people died. It wasn't that pathogenic. The one group of people who died in disproportionately large numbers were pregnant women. So pretty much everyone would be okay if they got infected, except for pregnant women. Why that is, no one knows. It's one of the mysteries of flu. So if you have these signs, then there's a good chance that that last slide has happened. So the flu's infected your cells. It's spending the cell's resources to make more copies of itself. You feel like this. You have all these symptoms and signs. But what's going on inside your body is shown here. And this is the part I really love. Like when our immune system and pathogenic microbes come together, if you look at that from a broad point of view, it's an amazing process because we have mechanisms to fight this part of the pathogen. They have mechanisms to fight back. And so it's kind of an overplay. And if you look at like a long-term evolution, like they evolve a way to do this, so we evolve a way to fight it this way, and then they evolve a different way to do this. And so it's kind of this never-ending battle. But in general, here's what's going on with your immune system. And this first part is probably the most important part. This is a microbe. It could be a virus, it could be a bacteria, it doesn't matter. Story's the same. The important part is on the outer surface. We're going to call it the flu just to be consistent. On the outer surface of the flu here, we have proteins that stick out. So they're coating it. These are not proteins that are inside, they're outside of it, right? So our body can see them, right? So if you think about the flu, it's different than us. It's not human, right? So on the surface of the flu virus and any other virus are going to be proteins that we don't have, okay? So our immune system, it has to decide what to kill and what to leave alone all the time. And so our immune system is trained to recognize every single protein we make. So if an immune system cell comes along, finds one of our cells, it's going to recognize it and leave it alone, right? But if something comes into the body that has proteins on the surface that our immune system cells knows are foreign, then that thing is going to die, okay? So that's what non-self is. Anything coming into the body, even if it's fake, if it has a surface structure that looks different than our cells, our immune system knows it, and our immune system is going to try to kill it. So what happens is, this thing comes into our body, our immune system says, hey, there is something here that doesn't belong. This is non-self. I know these shapes on the outer surface aren't us, so we're going to kill it. So what happens is your body makes antibodies by the billions. So we make these. These are the good guys, okay? These antibodies are extremely specific. They're going to bind to one thing and one thing only. You see how the end of this antibody has little circles here? I employed some PowerPoint trickery to try to make them circles. That means these antibodies can bind to this circle structure, and that is it. Antibody won't bind to anything else, right? So I tried here, not as successfully, to make these antibodies have a little V shape. So those antibodies are going to bind to this, right? It's like a lock and a key, right? The key has to slide into the lock for anything to happen. Same thing with an antibody and the proteins on the surface of any organism. The antibodies have to bind to the outside proteins on the virus for anything to happen. And if they do, then you get this. So this is our virus. We made these antibodies to it, so now they bind to our virus. And here's another one of the great truths. If an antibody in our body binds to something, that something dies. We're not going to go into the mechanism of how, but that's true. If the antibody binds to anything, that something dies. That's why these antibodies have to be so specific, because if they bound to our cells, then our cells would be killed, right? So the body makes very, very specific antibodies that bind to whatever came in, and that results in it being killed. That's called a primary immune response, okay? This is a very, very effective way to kill an incoming pathogen, but it has one trait that makes it kind of a bummer sometimes, for lack of a better word. And that kind of thing that makes it hard to be effective is that it takes weeks for this system to fully come online and be as strong as it can be, okay? So if you get infected with a virus and your body needs a couple weeks to get a full immune response to it, but the virus kills you in 3 days, it's not really that much of a help, right? But one really good thing about this primary immune response is that it has memory. So if you get sick from something and then you recover, all those resources that your body made to fight that one specific pathogen don't go away. What your body does is make an entire army of immune system cells that recognize just this pathogen, right? And if you're ever infected again, maybe a month later, 20 years later, it doesn't matter. You have an army of cells that are going to attack this pathogen. And so what happens is this. I know graphs on a Saturday morning. It's like, but here's the main point of this. If you look at just this left-hand side, this is our primary immune response. This is what happens first. So if you look at the y-axis here, or this line here, this is basically the strength of your immune system, right? So the higher you go on the line, the stronger your immune system is at killing something. Down here on the x-axis, we have the passage of time, okay? So like on the last slide, virus comes in, our immune system starts to fight it, and 10 days after fighting it for 10 days, we have this level of immune response. But it keeps building, right? So 30 days later, we have a much higher level of immune response. Now, this is okay unless the virus killed us at day 4, right? If we survive that initial infection, then we have that memory, that immune system memory in place. And so if we ever get infected with that exact same, has to be the exact same virus ever again, things are completely different. So now here we're at day 0. We get infected with the exact same organism. Look what happens at day 1. Our immune system is through the roof immediately, okay? So if you get infected with something naturally and you get sick and then you get better, there's a really good chance you've been infected with that many times during your life and you don't even know it because your immune system killed it so quick that you didn't even have time to develop symptoms. You know how little kids get sick all the time, right? As you get older, you don't get sick like that anymore. You're still getting exposed to the same bugs, but during your childhood, you're going through this process. And so later on, when you get exposed over and over again, this is the process your body uses to kill them, which is so much faster. Okay? So if you can survive that initial infection, then if you're ever infected with that same thing again, your immune system is way better at fighting it because it already knows how. And instead of having just a few cells that can attack it, it has an army of thousands in reserve just waiting for that one thing to come back. Okay? So this is what happens in real life. Flu comes in. You start this initial response, right? Sometimes you live. Most of the time you live. Sometimes you die. Right? If you do live, then you have this army of memory cells in reserve that can help you if you're ever infected with the exact same virus again. A lot of times people lose. People lose the battle with the flu. And so if you look on average, this is for the US. So this is an average number, and so some years it's probably much lower, some years it's much higher. But if you look at the data for the last few decades, we know that every year in the United States, about 36,000 people die of the flu, okay? You put that in perspective and think about society, it's kind of interesting. Like, if you read the news nowadays, Ebola's all over the news, right? How many people have died of Ebola this year? 1, right? So 36,000 people are going to die from the flu, roughly. And it doesn't get nearly as much attention because we're so used to seeing it year after year. But it kind of puts the Ebola thing in perspective. Ebola is much harder to get, much, much harder to get. And compared to flu, it doesn't cause nearly the damage. Okay, so this number is unacceptably high, right? We don't want 36,000 people to die every year from the flu. And also, we know from 1918 that every now and then there are flus that kill a lot more people than 36,000. Right? In 1918, it was almost 700,000. So what we want to do is come up with a way to teach our immune system how to fight the flu before the flu ever actually infects us. And that's what a vaccine is. What a vaccine tries to do is skip this part naturally. Make a vaccine that makes your body go through this process. So if you're ever actually infected with the real flu, you fight it this way, which is going to drastically increase your chances of survival. So here's how we do that. Same picture, right? So virus comes in, virus infects us. Same old story. The only thing our immune system cares about is the proteins on the surface of the cell. So if you think about that, what's going on inside of this virus, the immune system doesn't care about. The only thing it cares about is, can it recognize the proteins on the surface? Because if you can kill these, you can kill the whole organism, right? So what we do in one way or another, this is kind of a simplistic way to do it, but it's pretty accurate, is we take the flu virus and we chop it up into pieces. We treat it chemically with all this stuff, goes through this whole long process, but this is basically what it does. You take the flu virus and you inactivate it. Now, two important things happen here. The proteins on the surface are still there, so you can teach your body how to fight those, right? The other important thing is this thing has been destroyed as far as activity goes, so it cannot make you sick, okay? So what we do is we put this inside of this little bottle, and then we shoot that into your arm, and what your immune system does is just like before. It sees these molecules right here and says, those are not me, those are non-self. So it mounts an immune response to them, makes antibodies to fight them, right? And so we have these antibodies in our body. And then if we're ever actually infected with this virus, we already have the antibodies in the cells in place, so we can immediately kill it, okay? The only difference is when we use this method, if we back up a little bit, we don't have to go through this, because the vaccine taught our body how to fight that one virus or that one bacteria. And so if we're infected with the real one, we get an immediate and very strong immune response, which is going to increase the likelihood of us surviving that infection. It's going to make it a lot higher. Now, when it comes to making vaccines, some viruses are easy to make vaccines to, some are hard, and some are impossible. And whether or not you can easily make a vaccine to something has to do with how quickly the virus mutates, okay? So we all got vaccinated for polio, right? There's 3 kinds of polio that live out in the dirt somewhere, spread there by fecal matter. Polio doesn't mutate, right? So it never changes the shape of its outside proteins. So once you're vaccinated for those 3 kinds of polio, polio virus doesn't change shape, so you're good to go forever, right? HIV virus, it changes the shape of its outside molecules every single day, right? So you could easily make a vaccine for HIV. The problem is the antibodies you would get from that wouldn't work the day after, right? So if someone's infected with HIV, especially if they're not taking drugs, one cell goes in, thousands and thousands come out, and the virus that you have tomorrow looks nothing like the virus you have today. It's still HIV, but it's mutated to change shape. Okay? So we have polio, easy. We have HIV, impossible, at least so far. And in the middle, we have hard. One of the hard vaccines to make is for flu. And the reason it's hard to make a vaccine for flu is that it mutates as well. Not as fast as HIV, but it does mutate. So here's what we do. Well, if we look at this, let me explain that a little farther. So this is the same influenza virus we've been talking about, right? It looks a little different just because it's a different picture from the internet. But here we have our H proteins, hemagglutinin. Here we have our N proteins, our neuraminidase. These are the proteins on the outside of the virus. These are the ones the immune system is going to try to learn how to kill, right? Inside here, this is only showing 2 of the blueprints, the genome, but we know there's 8, right? And so what happens flu is this. Most of the time, we have like seasonal changes in flu, right? You got to get a flu shot every year. And why is that? This is why. If you look at the structure of flu, every year it's mutating slowly, right? That's called antigenic drift. And what that means is these blueprints are changing just a little bit. Like a mutation happens here, there. The virus changes the shape of its H or N proteins just a little bit. So this could be the flu virus this year, and the H proteins look like a perfect triangle. Because it's going to slowly mutate before next year, maybe next year the H proteins look like half a triangle. It still looks pretty close, right? But the flu vaccine this year is probably not going to work that great for next year because it's all about shape, right? The antibodies have to bind to that foreign invader, and that's going to kill it. So if the antibodies don't bind very well or not at all those immune system cells are different. So the ones you made last year to fight the flu may not work this year. That's why you got to get a flu shot every year. That is normal. So, and this usually is not very pathogenic at all, right? What happens once in a blue moon or once every 30 years or so on average is something entirely different. This is called antigenic shift. It still is all about the shapes on the outside of the virus, though. But what happens is the virus completely and utterly changes. So if you look here, here's our H protein, right? Looks like a triangle still. All of a sudden, something would happen inside the flu virus to make it mutate so much that it would change to an entirely different shape. Okay? So this virus is utterly and completely different than the one that existed just previously in time. So our immune system has no chance of dealing with that. Okay? So this usually causes people to get sick. This is what causes pandemics to happen. Pandemics are infections that happen worldwide. Okay? Like an outbreak would be if everyone in this room got sick. An epidemic would be if like everyone in the Southwest or something. A pandemic is all over the world. Okay? So because of this fact, so if we compare this to polio and HIV, which we talked about, if polio looked like this, it'd look like this next year and the year after and the year after and the year after. It would never change. So we'd never need a new vaccine. If we talk about HIV, it would change from this to this tomorrow, not months from now, tomorrow, and the next day it would look completely differently as well. So it's all about the shape on the proteins on the outer surface of a virus that are going to dictate how well your immune system can kill it. And flu is constantly and slowly changing. So here's how we try to deal with that. If you look worldwide, the World Health Organization has 115 different locations in 83 countries in the world. And the whole purpose of these little institutions is to keep track of what kind of flus are working in their area. Okay? So all those people keep track of what flus are prevalent in their area, and in February they get together and they have a big meeting. And they decide which 3 flus are going to be the biggest problem 6 months later. So they're guessing, right? So if they have a flu that's really pathogenic, super pathogenic, they'll probably stick that one in there, right? But either way, it's a guess. So in February, they pick the 3 flu viruses they want to make a vaccine to. They inject them into eggs. They inject them into eggs because it's really hard to grow viruses in a lab, but we do know from past experience that it will grow in eggs. So we grow the vaccine up in eggs. Once the eggs are filled with flu viruses, we break them open, purify all the flu viruses out of the eggs, get rid of all the egg stuff, take the viruses, inactivate them, or chop them up like we looked at a few pictures ago, put them in this bottle, and then we send them to Safeway and other places. By the end of September, and then in end of September, October, you start getting your flu vaccines. So good news is we can make flu vaccines. Bad news is it takes months to do it. And the other somewhat bad news is we're guessing, right? We're guessing which flus are going to be the problem months from now. And if you look at historical records on how accurate the guesses are, they're accurate about 75% of the time. Good solid C, right? They pass, just barely. So what that means, though, is 25% of the time, the flu shot isn't going to protect you for the flus that become prominent months from now. But on the plus side, 75% of the time it will. So that's a decision that everyone has to kind of make for themselves. Have I really been talking for 55 minutes? Sorry. Okay. So what I want to finish up on here is when the bad things happen. So if we back up here, this is okay. The flu mutates a little bit with antigenic drift, looks basically the same next year, so our immune system is kind of okay at killing it already, but a new vaccine will help it. What's really bad news is when the influenza virus changes utterly and completely. That's when pandemics happen, and that's when lots of people die. So how does that happen? That's what we're going to end up here. So what you see on here is basically people, birds, and pigs, right? And we also have our flu virus. So same old story. The H and N proteins are on the outside. We have our 8 genome segments, or our 8 pages of blueprints on the inside. And so what happens is a flu virus can infect a person, and inside that one person, the flu can get a mutation, which is just one tiny change in one of the blueprint pages. So the flu virus is different, but really similar. That can cause either nothing to happen or an epidemic strain to happen that can infect quite a few people, but it's not really going to be that big of a deal. But here's what the really bad thing is. Influenza viruses also infect birds. Okay? So birds can be infected with influenza viruses. The avian or bird flus also have 8 different genome segments or 8 pages of blueprints. Pigs can also be infected by pig flu. The pig flu also has 8 blueprint pages for its genome as well. And when really bad things happen is when a flu virus, all of a sudden and out of the blue, gets the ability to infect a human. Okay? So if we back up and talked about how if the outside of the virus changed a huge amount, that was antigenic shift, that's how this happens. You have a flu virus in a bird that looks nothing like the human virus. And so if it gains the ability to infect a person, then the person's immune system, having never seen a bird flu before, has no chance. This is what happened in 1918. So in 1918, after that story I told you about when they actually found the virus, they found that it was basically a bird flu somewhere gained 2 abilities. It gained the ability to directly infect humans, and it also gained the ability to spread very easily between humans. Okay? That's happened in recent years a little bit, but what's happened is only half the story. Bird flus have gained the ability to infect people, but they haven't gained the ability to spread from person to person easily. So nothing catastrophic has happened yet. In 1918, the flu went straight into people. Their immune systems had never seen anything like that ever, and the virus was good at spreading from person to person. That's catastrophic. The other thing that also causes problems is when you have a pig. So pigs can be infected by pig flus, human flus, and bird flus. And every once in a blue moon, you get a pig, a little pig somewhere, infected by a bird flu, and a human flu, and a swine flu, all at the same time, right? And then when you're making new viruses, you need 8 blueprint pages, right? So you take a few from the bird flu, a few from the human, and a few from the pig, and you get this new virus out that's completely and utterly different than anything the human has ever seen before. So that's the other way that pandemics can happen. You completely reassort the 8 genome segments or the 8 blueprint pages inside the pig, and that new virus, if it infects a person, is unlike anything that their immune system's ever seen before. And that's happened once, and it's happened way more than once, and it's going to happen again, just the way it is. So if we look here, we talked about the 1918 flu. This was when a bird flu learned how to directly infect a person, and the virus learned how to go very effectively from person to person. So this killed somewhere around 50 to 100 million people. In 1957, we had the Asian flu. That should be Asian. The Asian flu killed about 2 million people. Okay? And that was a reassortment one as well. In 1968, we had another outbreak that killed about 1 million people, and that was from viruses that had recombined from different species as well. And so if you look at this slide here, it was made in 2006, which was a while ago, and so they're theorizing there's going to be another pandemic someday of all these viruses mixed together, and we know this has already happened. There was a pandemic in 2009. The nice thing about the pandemic in 2009 is it spread all over the world, but it wasn't that pathogenic. Unless I mentioned earlier you were a pregnant woman. It was devastating to pregnant women. So in 2009, that pandemic killed about 250,000 people, something like that. And so the take-home message from this is this first came to the forefront or brought our attention to flu in 1918. But this continues to happen throughout time, and it's going to happen again. We have the capability of making flu viruses, but the problem with them is they take a long time to make. And so if another pandemic were to show up like that, we could start to make a vaccine, but it might be too late. In 1918, the flu came for 4 months and was gone when it was really bad, and it takes longer than that to make a vaccine. So what people do now is put a lot of faith in the World Health Organization. They're constantly looking at these flu viruses coming out in different areas of the world and trying to predict which ones are going to be problematic. And those are the ones that they're hopefully going to make vaccines to. It's also why these flu viruses that kill people pretty effectively tend to show up in China. China has wet markets, right? A ton of people packed together with a ton of pigs, packed together with a ton of birds. That's a recipe for disaster, right? So we'll see what happens. Yeah. It's a bright little girl. So 15 seconds to spare. All right. So I don't want to cut into Sal's time. Anyone have any questions, comments? Yeah. So Ebola hasn't been studied that much because it's really hard to do. But the understanding now is that people who have Ebola and have recovered from it, they are most likely immune to it unless Ebola starts mutating a lot, which we don't know if it does that yet. So if you look in Africa, I was listening to a story on NPR, there's a lot of people some people who've recovered, and now they go back and work in those quarantine tents or whatever, because they should be totally safe, should be. But there's a lot of unknown things about Ebola. So one thing we do know is you can't get Ebola unless, you know how like the flu, it drifts around forever and you can breathe it in and it lives on surfaces for quite a while. To get Ebola, you basically have to come in contact with body fluids. So who gets sick are family members and medical people. Like if someone came up and like grabbed a hold of your ears and like coughed in your face, you could get it that way, but it would take something like that. It's not nearly as transmissible as the flu, thank goodness. So. Yeah. So that's a really good question. And so vaccines are not without their risks. So there's a couple different conditions you can get. And if you look at the rate of complication, 1 out of every 1,000,000 people that get a flu shot is going to die from it. 1 out of every 10,000 people who get the flu is going to die from it. So that's the decision people have to make on their own. There's very definitely risks associated with vaccines. I mean, you're putting something into your body. So you could get an infection in your skin just from penetrating inward, like if you scratched yourself with anything else. Sometimes people have severe reactions to some of the compounds in the vaccines. Sometimes those conditions that you just described, like the neuromuscular disease, happens, and no one knows why. The rates that they happen are, like I said, 1 in 1,000,000. So you very definitely can get a reaction from a vaccine. It's extremely rare. So it's like an opportunity cost. Yeah. Oh, you don't get a flu shot, sorry. Yeah. So once in like a very infrequently, you can have the virus leave the respiratory system, because what it does is that you get it, right? And it goes into the epithelial tissue of your respiratory tree. And it's possible that it can go even deeper, very rarely, and get into your blood. And there's some evidence that shows it can infect some of the neurons in your brain. And so that could cause that to happen. That would be almost, but not quite unheard of. So they can disseminate. Very rarely. Your body does a really good job at keeping things walled off. So, yeah.
I'm curious about the average duration.
Oh, that's such a good question. So first answer, I have no idea. Second answer is it depends on the person, and we know it lasts a long time. So kind of some anecdotal evidence. For vaccines in general. So we stopped vaccinating for smallpox in the early 70s, like 1971, 1972. So if you have that scar, right, you've been vaccinated for smallpox. We quit that in the early 70s, like 71, 72. I was born in 1974, so I don't have that, right? Now you flash forward to like 2003, right after 9/11, the lab I was working in, we started working for the Department of Defense with like anti-bioterrorism vaccines. And so we were working with smallpox. Or not with smallpox, we were making a vaccine to smallpox. And so to do that, we used a virus called monkeypox, and that can kill you. So we had to get vaccinated. Okay, so I had never been vaccinated before, ever, because I was too young. I got vaccinated, and I had a horrendous response to it. It's been known forever that the smallpox vaccine that we would give all the way up until the 70s is really, really hard on you. Like nowadays, it would kill people with HIV. It would kill people with organ transplants. You need a really good, high-level functioning immune system to take care of it. Okay? So I got super sick. And everyone in our lab who'd never had the vaccine before got super sick. There were also some people in our lab who were in their 50s. And so they'd been vaccinated. And so they got vaccinated again, like, what would that be? Like 35 years later, they didn't get sick at all. You could tell the vaccine worked because you can see a little pock develop, but all the signs that we had, like super high fever, swollen glands, like shaking all night, like it was pretty horrendous. The people who had been vaccinated before had none of that. So that would argue that at least in that instance, in those people, it lasts at least 35 years. Now, everyone's different, right? Like some people, you can have like a whole family get the same sickness, and some people get super sick, some people get kind of sick, and some people maybe don't get sick at all. So our immune systems are all different. And if you want to look at it really simplistically, it goes back to shape. So if we all get infected with the same exact flu, maybe my immune system can make antibodies that bind that flu really, really well. So I'm going to kill it pretty well, right? So if you make antibodies that bind to it but kind of weakly, then it's going to take you longer to kill them. So the answer is, my second answer, it's going to be different for everybody in every infection, but it can be multiple decades. So, yeah. Yeah. Well, no, it's not polio. It's a virus that, it's an enterovirus, right? So it's infecting inside your intestines. That's what polio did too. There's a lot of different viruses that it can infect that area. So it's similar in some ways. It is not polio. It's not polio. Yeah, because it's an enteric virus. It's one that's going to attack and infect your digestive system basically, and so at least initially. And so that is going to come with a certain set of signs and symptoms that's going to be similar to polio because it does that too. But it's not polio. So, yeah, so far at least, you get vaccinated for those 3 kinds of polio and you were good. The fascinating thing about viruses is this: like if you look at humans and trees and bacteria and whales and chickens or whatever you want to talk about, we all use DNA for our blueprints, and we all do things pretty much the exact same way when it comes to building things. Viruses, you can throw the handbook out the window. So the blueprints of an HIV virus or the genome or a pox virus or the enterovirus, they're completely different. And they build themselves completely differently. So depending on how you build yourself, it comes with a certain set of kind of implications. Pox viruses, the way they build themselves, they have to be perfect. And so they don't mutate that much. HIV is about as sloppy as you can get. Like it makes a mistake literally every single time it mutates. Statistically, it makes a mistake every single time it makes a new virus. That's why you have completely different families of viruses the next day. So those things are going to dictate how well vaccines work, and they're also going to dictate how the virus spreads and what it's capable of. And so polio is one of those ones that's stable. Smallpox, stable. Polio, did I say that? Stable. Some of them change a little bit, like that's like flu, unless there's an antigenic shift. And then there's ones like HIV that are, am I going over my time? If you get me going on this, we'll be here all day. There's ones like HIV that change every single time it replicates. So, did I answer your question? I forgot what it was. I went kind of down there. So, yeah.
Early on, you said you weren't going to go into the way the virus dies, but you said if the body attaches, the virus dies. My question is, and I'm not sure, the function of the changing of the shape, so the virus does not get into the cell anymore.
So that's one thing antibodies can do. So if you have a virus floating around that's not in a cell yet, then those antibodies bind to H, right? And then the virus needs H to get into the cell. So antibodies binding to H before it gets into the cells is like wrapping a key in duct tape and trying to get it in. It doesn't work, right? Same thing for the virus. I cut out a lot of detail just for time, but so the picture I have shown, if you have a virus and you have an antibody stuck to it, like antibody, antibodies have a region called the FC region, and I know you know a lot about cells. Macrophages bind to that, engulf the whole thing and kill it all. So they work for free virus and virus getting in. So, okay, I got enough is enough. Okay, thank you guys.